All right, welcome back. We are now going to explore into diagnosing infections. So we need to understand aspects of the microorganism or the invader, the infection, so we can try to diagnose what's going to happen. And we involve a lot of different things collectively: a history of the patient, uh, causative types of things related to symptoms, and there's a huge history of that, of course, the doctors do. And eventually, artificial intelligence is going to have a huge impact. But for right now, what we're going to do is just to follow along and look at the diagnosing infections uh, in this chapter. So we're going to list the major categories of the identification technique and then provide a sentence description for each of these categories as we go along. This is not a involved chapter, and I'm truncating it so that there's certain things that I don't want you to worry about following. So I'll tell you clearly what those are. So that's probably good news for some of you. Uh, so how to identify bacteria in a patient specimen sample, and this follows along pretty much everything that this course is about, and it's a good time to discuss that. We're going to look at the phenotypic type, considers the macroscopic and microscopic morphology, physiology, and biochemistry of the bacteria. The immunological is look at serological type of analysis that we can do, or analyses, and the genotype is genetic techniques. Now, I'm not really in position to go into too much of the genetics. The folks that are in power that for this course, we're not really going to cover much of that. I guess you'll get that at a later date, but it's not really part of our objectives. So I'll let you know what we won't need to cover for right now. So with these types of identification, the phenotypic, immunologic, and genotype is really just looking at the basic microbiology, the microscopic, macroscopic, if what appears at the colony level, and looking at some of the just physical biochemistry. So those are just running the regular tests like the McConkey's or the Simmons citrate. It just goes on, and look at those characteristics. Then you look at the immunological. In other words, let's run a strep A test, and that's a latex layer test. Easy to do. We do that in the lab all the time. It's straightforward, and you can look at that. Or if something if you have an immune response, in other words, has the patient seen this, you'll see agglutination and things like that, which means like red blood cells will kind of pile up and becomes clotty or congealed, where if it's negative, it doesn't. So there's some immunological tests that we can run. And then, of course, the genotype is that we look at the genetics, and we won't go into too much detail on that. But the genetic means of identification are being used as a sole resource for identifying bacteria. So it helps at the point of care diagnosis because it's so doggone quick. You don't have to send them out. Other techniques that can assist in diagnosis, in addition to all the others that we talked about, is the computerized tomography, MRIs, positron emission tomography, or PET. These things that we can use to look at the swellings or various changes that we might predict with our initial sort of presumptive value that we might see. Presumptive being that we think we know what it is, we're just going to start confirming it, unless we need to throw out the window, of course, our initial diagnosis because we were wrong, which happens. That case study that I sent around, there was a doctor involved in that case study that was wrong, and he had to admit it and move on. And it happens, and that's, it's better to be open-minded and to change and do it right. So observation of traits that an organism is expressing, we need to be truthful and not what we think we should see, but we understand and analyze what we actually do see. We have to examine the appearance and behavior, looking at the enzymatic activities. If we run some, what we run, the conditions that it thrives at. It does better at 42 degrees and 37 degrees. It tells us something. What antibiotic profile it has. In other words, what's it susceptible to. Chemical composition of its cell wall or membranes. It's those things that we can do. And it's like anything a detector does. It's a preponderance of the evidence, and it builds a case. And the more evidence that we generate, the better our case will be. So antibody response is exploited for diagnostic purposes when a patient sample is tested for the presence of a specific antibody to a suspect. In other words, if the patient has seen it, then they should have an immune response, and we can test that with an agglutination type test. It's even easier than testing for the microbe itself. In other words, you just put the microbe in the presence of blood or serum, and then you see, and it kind of tells you if the human has seen those antigens before associated with that pathogen. Laboratory kits are available for immediate identification, so it goes through and really kind of automates the whole process. So sometimes some labs will just screen right through it. So that's nice. The advantages of a genomic method, of course, is you don't have to culture it. It's culture free. So you look for the presence of the genetics. So genotypic methods are increasingly automated, producing rapid results and more precise. And it's very sensitive, parts per billion, or even better. And it's viable, non-culturable microbes that cannot be grown in a laboratory, but still there's enough genetics there that we can pick it up, and that's valuable. Otherwise, clinically, we wouldn't have a clue. A gram stain of a sputum, in other words, someone's mouth contents from a patient, is an example. What type of method? So we're going to do a genotypic route of this. Are we going to look at immunologic, phenotypic, histologic? Well, this is hard. You really, and this question's kind of tricky, so I wouldn't ask this, but you look at it under a microscope. So technically, the answer would be none of the above, but histologic might be construed or could be viewed as a microscopic view, and that in itself could throw off. So I wouldn't put a question like this, but the idea is that you need to identify it using a microscope, and if you call it histology or not, typically that's used just for tissues, not looking at gram stains. But anyhow, I thought I left this in here just to make that argument, so that I try not to add ever tricky questions, but sometimes I miss them or I don't interpret that as a potential tricky, and this falls into that category. Histopathology is the branch of histology that includes microscopic identification, a study of diseased tissue. So I worked in pathology at one time and looking at changes in cellular makeup of tissues. That can be quite challenging. It's an important part of any anatomical pathology and surgical pathology. Looking at these sorts of things, cancers and other types of diseases, or effects of toxins, histotoxicology, and those sorts of things can come out. But histology is one of those things that it takes a fair amount of time and experience to go through, slides on a regular basis. It's really time consuming. Okay, so now that we're going to talk about identity factors that may affect the identification of an infectious agent, and then we're going to compare the tests. So again, it's a preponderance of the evidence. Specimen collection. This is without a doubt, I think, in some ways one of the harder parts, depending on what we're doing. But you have to use aseptic technique. You don't want to cross-contaminate or contaminate just because of means of collection. Those can really cause you to go down the wrong path. Sterile sample containers and other tools prevent the contamination, but anytime you add a patient into the mix and they need privacy in the bathroom, Lord knows what's going on. So just keep that in mind. Care should be taken with samples that contain resident microbiota, and only the infected site should be sampled, not the surrounding areas. And that's a pretty tall order, to be honest with you. So the certain types of sampling: sputum, swabbing the throat, getting close as you can down the trachea; blood samples, urine samples, and that's a problem because men and women are of course different, and clear catching urine in females a little bit harder than in men, but men tend to not understand what have a stream already established, and then you start collecting versus the early stream, and then you get all sorts of things that the urethra might have contained. So the clean catch is important. Catheters, you want to collect those, but unfortunately those can get contaminated on the way in. Skin swabs, you got to be pretty good to know where those things where you need to go and get the samples. So anyhow, the idea behind it we need to know about is like feces and the vaginal swabs and things. There's certain techniques, SOPs, that you follow for that, and it'll all come out at the end. Oh well, I shouldn't have used that analogy. It will work. Spinal taps and things are SOPs. A lot of times when you do certain types of specific type of sampling, you might use the assistance of some kind of visualization. There's that transport swab I was talking about. There's the cap, and then there's the swab, and then there's a liquid at the bottom, and you usually squeeze it to break a seal on it, and so that it keeps the end of the swab wet. Desiccation can kill. So this is a good slide. I mean, it kind of covers all the areas and things that one has to worry about. Sputum samples discharged by coughing are collected with a catheter. Contamination with saliva should be avoided since it contains millions of bacteria, but that's pretty much the way it is. So a sputum will contain saliva. That's why I always say when you kind of cough it up, they spit it out. That's kind of how it is. The saliva sample is obtained by having a patient expert into a container, and the idea is you want to get the mass up from the lung that's being communicated up through the cilia action of the trachea, but these two are one of the hardest ones to delineate, so that's kind of how it is. Urine samples can be taken aseptically from the bladder with a catheter. Clean catch is you usually wash the external urethra, but in the male, of course, you have that longer channel that washes or flushes, so you want to go ahead and start the stream and then collect, and it's an interrupted stream. But the problem is you get other pathogens in there. They call it a dirty catch. And the first voided urine is required for diagnostic techniques. For some reason, you may want to see if there is a buildup of a particular type of pathogen in the lining. Mucus linings of urethra, vaginal, or cervix may be sampled with swab or applicator stick. Pretty straightforward stuff. And SOPs for all of it. Skin samples, swab, or scraped with a scalpel to expose deeper layers. Wounds are sampled with swabbing with a punch biopsy type of tool, or a lavage. It's usually going in with a liquid and rinsing it and then collecting it back into a syringe. And so that's kind of where we are with that. So I guess that covers pretty much antisepsis of the puncture site. Of course, you want to make sure it's as clean as it can possibly be before you take the sample. Other specimen sources are the eyes and ears and synovial fluid of joints, nasal cavity, all sorts of proper collection and labeling and identifying. So you may see barcodes being used. It's a way of distributing lots of different tags, and it's easy to read with all sorts of barcoding equipment. That way we don't lose the right type of treatment and identification of the patient and samples taken from the patient. It would be terrible to have a misdiagnosis due to sloppy type of paperwork. And then Uh, Overview of the laboratory techniques, patient analysis for signs of infection, and all sorts of things. Usually picked up in triage, followed up. But you see, I use a form like this in the lab for students to get experience in writing sort of a typical report that it does these types of culture results, and then looking at antibiotic sensitivity down here, and then you have your analysis and various things here. And then the most important part is that you have to sign it as a technologist, and in other words, you're putting your name on it, so you want it right. You got comments to try to qualify what the identification is, and the task is to be as right as possible, and you want the preponderance of the evidence to be in your favor, and that's kind of what the clinician has to worry about. Care should be taken to avoid contamination with what when taking samples from a patient. And the problem that is the normal microbiota, I think, is what you would think, and that is the best answer is you want to try to avoid that unless you're looking at that. So it depends. The answer technically would be none of the choices is correct. But again, this is one of those tricky type questions of being normal microbiota. You don't want that, so you want to kind of clean that area, sort of like cleaning the end of genitalia and stuff, and then getting urine sample. Then that would get rid of the normal microbiota. So you want to avoid the contamination with the normal, but none of the choices really fit that one. But I wouldn't give those type of questions on an exam. List 3 different tests that fall into the direct category, and then 2 drawbacks of phenotypic testing methods. And so these are easy. What we look for, the direct observation, we use the gram stain, and that's used pretty much all the time. I can do a gram stain pretty fast within the limits, and I'll have a result probably in less than 20 minutes if I need it. And if it doesn't stain, or it's not, it doesn't make sense, then I move to an acid fast, because then we're worried that we're looking at tuberculosis or something else. But the symptoms would be there. But you can see the 2 stains. This is a mixture of both gram positive, gram negative, the gram negative rods, and the gram positive cocci you see there, and then of course the acid fast. You see the bacillus sitting in there. Pretty standard, easy to do. So we have the specialized media used to enrich pathogens in small numbers, selective media used for non-sterile, containing diverse of bacteria species to encourage growth. So if we're looking for gram positive, gram negative, and differential, of course, we've already talked about all this. We can use that. A dichotomous key is a tool that allows the user to determine the identity of items, and usually we can look at a branched sort of, if this is true and this is true and this is true. That's a dichotomous key. So you look through and it kind of branches until you find the answer. So here you go. The idea is it's gram positive, it's catalase positive, it's strictly aerobic, it's got to be a micrococcus and that sort of thing. And you would run through a dichotomous key. And I provide those when we look at identifying unknowns. I don't have you memorize those. It's always a lookup. Biochemical testing, physiological reactions of bacteria to nutrients and other substrates, provide evidence of enzyme systems. All sorts of these tests you can buy. They're out there. Enzyme-mediated metabolic reactions. You culture in a medium with special substrate. Then you test it by putting it in some kit, and then you see the enzymes being made visible by some sort of dye, and no coloration means it lacks that particular enzyme. So we can do that pretty quick, and if you do the enzyme, the product's formed or it's not, and then that's part of the preponderance of the evidence that we use. And so we have batches of these things, kits that we just usually put it in a solution, a growth media, and it's been inoculated, so the bacteria is there, and I just apply it across the board, wait a certain amount of time, and then I read it, and so you get a positive here, and here, and you enter it usually into a computer program, and it kicks out Edwardsellia tardia is the organism. It makes it pretty quick, and you can have your answers in about 6 to 8 hours. Mycobacteria growth on the indicator tube, they call it MG or what they call MGIT. It detects the growth of the slow-growing Mycobacterium tuberculosis, monitors oxygen levels in a tube. Silicon chip, so this is a system on a chip, so the detection is on a semiconductor surface or a wafer. The silicon chip at the bottom of the tube is impregnated with fluorescent substance sensitive to oxygen levels that begin to fluoresce as the oxygen levels in the media decrease. Then you know that it's utilizing the oxygens to slow grower like Mycobacterium tuberculosis. Automated machines will give a visual and audible signal if it's positive, so we can move on with it. So the phenotypic test, just know that the Mycobacterial growth indicator tube, MGIT, is a phenotypic type of test to look at oxygen levels. Antimicrobial sensitivity testing is really straightforward. It's useful in determining which drugs will be used in a treatment. We'll be talking about this in the lab at some great length. In crisis level rise in antimicrobial resistance, we see it across the board. We can't waste much time in trying to find the right and useful treatment path. Phage typing is, the phages are coming back. We have high resistance models, so we want to know what the results are. Bacteriophages can actually be used as a treatment. There are some products out there that the bacteriophages will destroy the particular organism. The problem is, just like with antibiotics, the bacteria have learned to become resistant to phages, certain types, and so that it still is an issue. But bacteriophage will infect the bacteria, usually very specific, often used for tracing strains, and it shows up on a plate by leaving a plaque. It's a very obvious clearing where the bacteria should have grown but didn't. So it makes it really easy, and usually we do it as a lawn of bacteria, and then you can actually count the number of plaques on a plate, and it's really easy. It's been around for a long time, and it's coming back and more use of it. And so you can phage type an unknown, and you put drops on there, and you see two areas that are cleared, so we know that particular phage. Where it's cleared, we know it was active. The other regions we don't. So bacteriophage typing is pretty common. Determine clinical significance of culture. It's important to rapidly determine if an isolate from a specimen is clinically important. A few colonies of E. coli in urine can be indicative of normal biota, but several hundreds can mean an active infection. So it's a common sense sort of call. A single colony of a true pathogen such as Mycobacterium tuberculosis and sputum is highly suggestive of disease. We know that that's usually not a good thing. Repeated isolation of relatively pure culture of any microorganism means it's most likely, you've nailed it, it's the pathogen. The drawbacks of phenotypic methods: a microbe has to be cultured. It takes a minimum of 18 to 24 hours. The genetic tests are a lot faster than that. Many infectious conditions can be caused by non-culturable organisms, leaving open the possibility that the organism we do culture is simply sort of there as a bystander, and it happens. So the direct methods are looking at microscopy, looking at it. You don't have to culture it. Biochemical susceptibility type of testing means that you have to culture it. Knowing if it's selective or differential type of viewings, again, you're looking at 18, 24 hours, and a lot of times we just don't have the time. So if you've obtained a fecal sample from a patient, what type of media would you grow it? And the answer for this is would you use a pathogen for the sample? And the answer for this one is all of them are correct, and that's pretty straightforward. We're going to stop here. I don't want you to worry about any other materials from 15.4 on. just hold you to 15.3 in your book. The rest of it is not part of the objectives. These few things I wanted to include, they are included in our course objectives, and I don't want to waste any of the other time, and this is pretty straightforward. So whoops, that will make our part of this a lot easier for the exam on coming up soon here. So this will be included. This is chapter 15, but only what I've talked about in this slide set, and I'll make this of course available to you. Alrighty, we're in good shape, and more coming.